Okay. Hello, everyone. So this is a project explanation for DNA analysis using image processing, and we are going to use something like gel electrophoresis images. Now, what is basically this thing is, that DNA is unique for multiple people, and you can find out whom you are genetically related with, like your father, mother, grandparents, and so on. So this has multiple applications. Now, tracing his DNA goes to something called as PCR. Now, because of the corona disease, you may say most of us have heard the term RT-PCR, which we call as reverse transcription PCR. But in general, PCR is polymerase chain reaction. And what it does is basically it has this DNA strand, which is like a helix, and it reopens them. So it splits down this DNA into two RNAs, and then it has a binding element inside it. So once it binds, it starts duplicating itself, and basically this chain reaction creates a multiple copies of this DNA so that it becomes a detectable. So you can imagine even a drop of saliva from your mouth can be used to produce a huge amount of DNA. And then once we have sufficiently long DNA, we use some process called gel electrophoresis. So we apply roughly 200-volt DC on both sides of the gel, and then DNA tries to pass through this gel, and depending upon the type of the DNA, it goes and stuck at certain point, and it doesn't go further. And once we see this gel under UV, wherever that particular DNA is, there will be a band appearing. So basically, our process starts from here. We'll have this gel electrophoresis images. Let me check if I can show you some on my screen. Uh, Okay, so this is the bands what I was talking about. Now, typical images may look something like this, and each of this band will have its corresponding value. And when you see a band at corresponding ladder, so this we call as a ladder. I hope my mouse pointer is visible. And each value in that ladder has particular significance about that particular DNA. So this is how a digital image would look like. So you have marker sample which creates this ladder, then your sample. So typically gel electrophoresis is run for multiple things, like here they are running for L5, L49, L57, L15, L62. And then if this is your mother's DNA, same is your father's, your DNA, your grandfather's DNA. So if everything is matching, you will see all the bands in the same line, and that corresponds to a certain number in the ladder. And similarly, you can find, so this is how the gel electrophoresis happens. This is vertical setup, but you can have this kind of horizontal setup also. These ones are more popular. And once you have these images, you have to apply your deep learning knowledge to extract the information out of it. To start with, a baby step you can do is go to a website called Google Scholar and search for something like DNA. Let me go to Scholar website, and then you can search for something like image analysis for DNA and so on. So try to put these kind of keywords and see what papers you get. Most of the PDFs you can download from here. If not, you can use a website which is called as Sci-Hub. And I don't know how many are there in this group. I guess one, two, three, four. So total 20 papers you will download for this particular project. And once you have these 20 papers, you will basically divide these papers. So you will have 20 papers combined. You can conduct another Zoom meeting where each one will get 5 papers each to read, and you will prepare a small presentation explaining what it is done in that particular paper. the presentation should be very simple, like on a white slide you put a black letters with Times New Roman font size 24, and one picture per slide is must. This picture can be from the paper itself to better explain the concept, and not more than 2 to 3 lines on a slide. So this is the task one, you can say, common for most of the groups. And then you go for the particular detailed task. So since you want to do some image analysis for this jail doc images, what I will say is you learn to install OpenCV on Python. There is one good tutorial on Instructable by myself and Mr. Amit. And then you can also try and download one gel image. Uh, so most of you, whenever you download, you create a separate folder which we can use for supervised learning later. So you download and name this folder for that particular gel image, whatever that gel image is for, and corresponding DNA number should be noted. And there could be different folders for different kind of images. If you get a big data set of gel electrophoresis images, nothing like it, you can start working directly. But right now I want you to take the baby steps. So just download one image, and then you will display it using Python program, and then do some basic operations like color to grayscale and so on. So this is the starting point, color to grayscale, and that will be your task one. So in the next meeting, we will discuss in details what is to be done, but this is how you will start. And I will stop this recording now. If you have any doubts, you can ask me.